Every 9 minutes, another name gets added to the transplant waiting list. But what if the solution to this crisis isn't human, but porcine? Pig organs are remarkably similar to ours in size and function, but there's a catch. Our body sees them as foreign invaders because of a singular sugar molecule called NEU5GC, produced by the pig's CMAH gene. This is where CRISPR changes everything. By editing just one gene, we can make pig organs compatible with human bodies. We designed a precise molecular guide to target the CMAH gene, like a genetic GPS that's accurate enough to avoid mistakes, but powerful enough to make the crucial edit. So, let me show you how we're going to be editing pig genomes to solve the organ shortage. For this video, we're going to be doing a hands-on genetic engineering using this software called Benchling. So first, we're going to pull up the DNA sequence of the CMAH protein. So if you just go to create right here, new DNA RNA sequence, click on that, import from database, search up CMAH. Genome, the scientific name for pigs is Suscropa. Technically, Suscropa is a wild pig or a boar. Suscropa domesticus is more of a pig, but that's besides the point. And then we click search. Then we should end up with perfect. We got the ENSSCG00001099 gene, which is exactly what we needed, and we're gonna go with the full genomic sequence, not just the coding regions, because CRISPR works best when we have the complete genetic content. We're just gonna go ahead and click import real quick. Slowly. Okay. And boom, there you have it. You have your imported CMAH gene. Now, if we just go and find exon 2, so we are designing our molecular scissors in Benchling's CRISPR tool. So I'm finding exon 2 of CMAH just because it's the perfect position to disrupt the gene when cut. And the best thing about Benchling is that it locates exon 2 for you. You don't have to manually input the start and end of it, which in this case would be 4,000 42,455 would be the start, and the end would be 42,546, but that's besides the point. Now we go over to the CRISPR tool, design and analyze guides, and we have the single guide chosen, and then we click finish. So as you see, the target region here is exon 2. They're the numbers for exon 2. It's auto-inputted because I have it highlighted. And our main goal here is to find a guide sequence that has a higher on-target score than 70-ish and a lower off-target score than around like less than 40, 50. And I think the perfect example right here is TCTTGAC blah 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 with PEM of AGG. So we'll go and click that and hit assemble. We're going to choose PX459 just because it's the most updated. And it already inputs the insertion region for you. So you just click next. You choose a folder for this to happen. I created a folder beforehand. So click assemble. Boom, your assembly's complete. Now, if you go through some of these other ones, you will be able to find see exactly what happened in your assembly. Look, right here it says your assembly is complete. Open the primers and the plasmids from the file browser at the left. So right here, you can also create a notebook entry, blah, blah, blah. So if we open the primer and the plastids, we get this, and it shows you more in depth of what actually happened. So you see the guide here, which is the CAG, blah, 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 but it's also present in this circular diagram of it, which is kind of similar to what this is, but a little bit different. So there you have it. We just completed a CMAH gene knockout successfully. We're one step closer to xenotransplantation. Thank you.